So it's a pleasure for me to be here with you and to present our work. In this presentation, I'm going to talk about Omic and meta-omic methodologies, and also about the works that we are developing at the University of Huelva in connection with metals, metabolites, and microbiota intertwined mechanisms. There is a connection between our gut microbiota and our brain that is very important for several diseases. For example, Alzheimer's disease, but not only neurological diseases, for example, also for cancer. And on the other hand, metals and other pollutants can enter the body through environmental pollution, and also, for example, essential trace elements like selenium can be present in food. And these metals can shape the diversity, the richness, and the composition of the gut microbiota. And then they can affect this communication between our gut microbiota and our brain. Also, there is another axis, which is the enteromammary gland axis, by which the mother transfers to the newborn microbiota. That is very important to see the infant gut. And human milk can contain also microbiota, but also it can contain pollutants. For example, mercury or arsenic can be present in food. And regarding selenium, this is a very important trace element that can be present in human milk, and it is involved in the thyroid metabolism. And for example, also persistent organic pollutants can be present in food, and it can be used instead of thyroid hormones because of the chemical similarity. Then all of this can impact in the gut microbiota brain axis of the newborn, the neurological development, and the reproduction, and for example, the hypothalamic pituitary gonadal axis. So breastfeeding is really a window of opportunity, and especially the 1,000 first days of life are very important for programming the future. And in this slide, we can see how is the gut-brain crosstalk. We have a direct communication between our gut microbiota and our brain, for example, through the vagus nerve, which is here, and that connects the brain with our gut. We have also an enteric nervous system. For example, we have this structure called neuropods that connect the vagus nerve. And also we have enteric glia here and enteroendocrine cells here. And also we have an indirect communication through neurotransmitter and neuroactive metabolites, for example, short-chain fatty acids or butyrate. We have also an communication through the immune pathway, through cytokines, and also through a neuroendocrine pathway, for example, the cortisol secretion through the hypothalamus pituitary adrenal axis. Due to the excellent properties of selenium, there is a wide variety of functional foods and nutraceuticals. For example, some time ago, we worked with Chlorella sorokiniana, which is a microalgae that can transform inorganic selenium into selenomethionine. And this is very important because for selenium, the beneficial role of selenium is related not only with the dose, but also with the chemical form. However, regarding formula milk, the Food and Drug Administration requires that infant formulas should be fortified with selenium, but it doesn't recommend a particular form of this element for the fortification. And recent research suggests that organic forms of selenium, especially enriched yeast, are more bioavailable, better absorbed and retained. and may have improved biological activity compared with anorganic forms. However, we need more studies evaluating the safety and health outcomes of fortification with organic selenium. And at the moment, for example, for human milk, only the fortification with anorganic selenium and selenomethionine is allowed. And selenium is very important also in pregnancy. For example, the thyroid gland is the organ with the highest content of selenium per gram of tissue. And we have specific selenoproteins, which are the diodes, the iodopyronine deiodinases, which transform the thyroxine T4, which is the most abundant thyroid hormone, into the most active 3-iodopyronine, which is the most active thyroid hormone. Then selenium is critical for fetal development, and selenium deficit can lead to problems in the mother and also in the offspring. And for example, the hypothyroidism can be used as a biomarker of selenium deficiency. And then selenium deficiency can lead to a decrease in the function of selenoproteins and a decrease in the synthesis of thyroid hormones responsible for the conversion of T4 into T3. In this slide, we can see the metabolism of thyroid hormone. We have a protein that is the thyroglobulin, which is a dimeric glycoprotein that is produced by follicular cells of the thyroid, and it is used entirely in the thyroid gland. It is used in the glands to produce T3 and T4, and also other thyroid hormones that are monoiodotyrosine and the iodotyrosine. Then selenium that we can see here is essential for the iodination of T4 by diodes, but also for the degradation of the excess production of hydrogen peroxide through GPX and thioredoxin reductase. And regarding the mother-offspring transfer of selenium, first studies in vitro human placental lobes suggested a mechanism of passive diffusion through the placental membrane. And years later, the affinity of this transporter was described for selenium and chromium oxyanions. And in Wistar rats, the availability and the mother-offspring transport of selenomethionine and inorganic selenium during pregnancy and lactation were compared. And these authors from Japan demonstrated that, except in kidneys, total selenium is altered in tissues and plasma during pregnancy and lactation. And during pregnancy, inorganic selenium and to a lesser extent selenomethionine are transformed into selenoprotein P. And this molecule is incorporated into placenta through its receptor ApoER2 during pregnancy. And in human milk, these molecules were previously identified: selenomethionine, selenocysteamine, inorganic selenium, and glutathione peroxidase. In the works I will comment today, we combine Omic methodologies that, as you know, are the massive determination of, for example, genes, genomics, that inform us about what could happen. Transcriptomics give us information about what seems that it happens, and proteomics about what provokes that it happens. And metabolomics inform us about what happened and what is happening just now. Metabolomics is the omics more closer to the phenotype, and metabolites that are small molecules with a mass below 1,500 Daltons, and they are the last mechanism of action and more closer to the phenotype. And in our works, we combine omic and meta-omic methodologies. Meta-omics are the massive determination of mixtures of genes, transcripts, proteins, or metabolites that come from several organisms. In our case, we apply meta-taxonomics, which is the determination of mixtures of genes, but not the entire gene, only this region, that come from several organisms. And the objective is to determine the microbiota profile in the gut content or in the human milk. And then we combine this meta-omic with heteroatom tag proteomics applied to human serum or human milk. And in this case, we first separate selenoproteins, for example, using size exclusion chromatography and affinity chromatography. But the important thing is that we determine the protein not with a molecular detector, but we use an atomic one. It is an inductively coupled plasma mass spectrometer. And then we use selenium as a tag. It is an heteroatom, that is an atom different to carbon, hydrogen, nitrogen, oxygen, or fluoride. And we use this element as a tag to detect the molecule. And then, for example, if we want to quantify selenoprotein P, as we know that selenoprotein P has 10 selenium atoms, we can determine selenium in the peak, and after that we can convert the concentration of total selenium in the peak into the concentration of selenoprotein. And also we combine this omic with untarget metabolomics applied to human serum, human milk, and also to the gut content. In this case, it will be a meta-omic. It can be called meta-metabolomics because we are determining the microbially produced metabolites that come from several organisms. And to determine metabolites, we combine gas chromatography with mass spectrometry with UHPLC-QTOF. And in this slide, you can see the main advantages and drawbacks of the different methodologies. In the upper part for the determination of the total concentration of metals, and here for the determination of metalloproteins. And for example, regarding the total content of metals, ICP-MS is a very good analytical technique. It is multi-elemental, it is rapid, it allows good control of interferences, allows isotopic dilution analysis for quantification, it has high sensitivity, and also you can determine the distribution of elements in tissues by coupling laser ablation to the ICP-MS. Regarding the drawbacks, it has a high cost of instrument acquisition and operation and maintenance. It is a high experience is required to work with ICP-MS, and the tolerance to dissolve solid is limited. For example, if you want to analyze urine or seawater samples, maybe you will have problems with an ICP-MS. And regarding the determination of metalloproteins, you can couple an HPLC to the ICP-MS for determination, and you can perform the absolute quantification of elements and chemical species. In this case, protein standards are not required because you are quantifying, for example, selenium. You don't need the standard for selenoprotein P. It allows the determination of phosphorylation of proteins. It has high sensitivity and also high accuracy and precision. However, for example, enzymatic activities cannot be determined, but it can be combined with metabolomics. And if you combine metabolomics with heteroatom tag proteomics, you can quantify the metabolism of these enzymes. And another drawback is that you need an heteroatom in the molecule to detect the protein by ICP-MS. And for example, Western blot allows the relative quantification of proteins, not the absolute quantification, and it allows the determination of the molecular weight. It cannot be performed with an ICP-MS. However, the drawback is that you cannot perform absolute quantification, and especially an specific antigen is required for each protein, and this is an important drawback. And for PCR, target relative or absolute quantification can be performed, but not for proteins. You can quantify transcripts. And maybe there is a possible lag between the transcript and proteins to correlate both. And only you can quantify DNA or RNA. And regarding spectrophotometry, you can determine enzymatic activities, and you can perform quantification of total proteins, and the cost is very low. However, you must combine several preparative techniques, and the sample volume consumption is very high. And regarding ELISA, it allows the absolute quantification. It is a simple technique. It is high specific. It can be automated, and it can be used as a diagnostic or forecast method. And the sample volume consumption is very low. However, as in the case of Western blot, the antigen must be well known, purified, and isolated first, and enzymatic activities cannot be estimated. And regarding the use of an ICP-MS or an atomic emission spectrometry, the reason is very simple. You can see here the determination of selenium ions in the lower part with ICP-MS. We can see here the mass 140 for this isotope, and also the mass 141. And the mass spectra is very clear. However, with an atomic emission spectrometry, we can see several interferences in the spectra. We have a continuous spectra here for the argon. Also, we can see here bands for molecules. So the spectra is very complicated. However, in the case of ICP-MS, we have also several signals that can be a problem. For example, the dimer of nitrogen for the determination of silicon, or this adduct for the determination of phosphorus, or oxygen plus oxygen for the determination of sulfur, argon oxide for iron, or this is a typical problem for the determination of selenium, argon plus argon. So the solution can be the correction with a blank, or to use another isotope of the analyte. However, we can use also a collision reaction ICP-MS system, in which we can eliminate the interferences by collision with a gas or by reaction. In the first case, we can use a modified quadrupole. In fact, it is an hexapole that is located before our analyzer, and we can introduce helium, and helium will impact with the interference, and it will eliminate. For example, polyatomic species are larger than the analytes, and the probability of collision is higher with the polyatomic species than for the analytes. And also we can eliminate interferences by reaction. For example, we can introduce hydrogen that will react with the analyte or with the interference. And in this slide, you can see different analyzers. The single quadrupole is very common, and also the triple quad here, the time of flight or sector field. And in this sense, the resolution increases. So, for example, for the determination of selenium, we can compare both systems, first with one quadrupole in the upper part, and here with three quadrupoles. So, if we want to determine selenium, the mass 80, we can introduce oxygen in the reaction cell, and oxygen will react with selenium to form selenium oxide with a mass of 96. And then we can detect this mass in the quadrupole, in the analyzer. However, as you can see here, we have other elements that has the same mass that selenium with oxygen, for example, zirconium or molybdenum or ruthenium. So we have interferences also after the reaction with oxygen. With a triple quadrupole system, we can do this. First, we can eliminate these masses after the reaction of oxygen, 96, in the first quadrupole. And after that, we can introduce oxygen to react with selenium. And after that, we can detect the mass in the third quadrupole. So we can eliminate completely the interferences of selenium. So this is the reason that for which the triple quadrupole system has high resolution. And in this work, we developed a method for the determination of selenoproteins in human serum. So we use this system in which we coupled two size exclusion columns to increase the resolution and two affinity columns. The first one to retain selenoprotein P, and the second one that retains selenoalbumin and selenoprotein P. This is the typical chromatogram obtained for human serum, in which, for example, the concentration of selenometabolites is very low, but we fortify the sample to see the separation between selenometabolites and glutathione peroxidase. And here we have the glutathione peroxidase peak. After that, selenometabolites, selenoprotein P, and selenoalbumin. And all these chemical forms of selenium accounts for about 90% of total selenium in the human serum. So our method can quantify the status of this element in the body. And also in the coupling, you can see that this column is in the column switching system, and it is isolated here because if we eliminate this, selenoprotein P and selenoalbumin will coalute in a single peak. So we need this column switching system to separate proteins. This is another method for selenium speciation, but only for selenometabolites. And in this case, we apply an ion exchange chromatography coupled to ICP-MS to separate selenometabolites. So this peak here, that is in which all the selenometabolites coalute in a single peak, here we can separate in different peaks. And here you can see inorganic selenium, selenium-4 and selenium-6, selenomethionine, selenocysteine, and selenomethylselenocysteine. selenocysteine. And here you can see both chromatograms compare. Here for selenoproteins, this is the real concentration of selenometabolites in human serum. And here you can see the speciation of selenometabolites. And for the validation of the methodology, we don't have a reference material. However, this material that is commercial, this reference material, is certified for total selenium, and several laboratories have determined the concentration of selenoproteins and selenometabolites. So we can use this to validate our method. And here you can see the typical concentration of these selenoproteins and total selenium in the reference material, and also for the selenometabolites. And an important thing in when using ICP-MS is that we can apply isotopic dilution analysis. In this slide, you can see an example for isotopic dilution analysis of thin isotopes. As you know, any sample in the world will have an a ratio between the isotopes that is the same. So this is a known natural ratio, and we can buy a standard enriched in one of the isotopes. So if I mix my sample with the standard, we will have an intermediate isotopic distribution. So we can apply this equation in which the only one unknown is the concentration of tin, because we can determine by ICP-MS the isotopic ratio between two masses with the ICP-MS, and the ratio in the sample is known, and also in the spike. So we can easily calculate the concentration of tin in the sample. So the isotopic dilution analysis has several important advantages. For example, the uncertainty in the concentration depends only on the isotopic ratio in the sample, in the spike, and in the mixture. Any liquid of the mixture has the same isotopic ratio. It is not necessary to know the dilution or preconcentration factors, and the equation does not have any factor related with instrumental sensitivity. And also, drift issues and magic effects are eliminated with this quantification method. And here you can see an example about isotopic dilution analysis. For example, if we want to know how many blue circles are here, we can mix with a known number of orange circles here, for example, 40. And after that, we can mix them, and we can select a part, and we can determine the ratio between orange and blue circles. So if we apply this equation, the only unknown is the number of blue circles here. And also, we can apply two different isotopic dilution analysis modes. Isotopic species specific. For example, if we add enriched selenomethionine, or in a specific species. For example, we can add post-column selenium with an isotope to quantify all the species with the same chemical form. And in this work, we try to apply the methodology for selenoproteins speciation in human serum to human milk. And it was not possible because the composition is different. So first, human serum can be directly injected into the HPLC, and human milk cannot be directly injected. So first, we have to separate the sample by ultracentrifugation, and after that, we obtain the fat in the upper part, and also the casein cells in the lower part, and in the middle, we can separate a serum from the human milk, and this can be directly injected into the HPLC. Here you can see the typical chromatogram of human milk, which is the red one, and also for the standards, which is the green one, and for human serum, but with a new methodology, which is the blue one. So in this configuration, we have three size exclusion columns which are required for to increase the resolution, and we have only one affinity column. And we need this column switching system because if not, selenocysteamine will coalesce with this peak because it has also affinity for the heparin in the affinity column. So we can see here that in the milk, the red chromatogram, we have selenometabolites here, glutathione peroxidase, selenocysteamine, and also we have a peak here that coalesces with selenoprotein P that has not been previously reported. So we isolated this peak and we digested with trypsin, and after that we try to identify this peak with mass spectrometry. And here you can see two different peptides obtained after the digestion of the protein. And especially this is very interesting because here you can see that it contains the isotopic distribution of selenium in this peptide. So we can see here that this is a selenium containing peptide. And then we identify selenoprotein P in human milk that was not previously reported. And this is very important because selenoprotein P is a transporter of selenium. It is also an antioxidant, but also is related with several neurological roles. And after that, we applied this method, this analytical method, to 20 couples of mothers and newborns. And we determined the different chemical forms of selenium in human milk, in cord serum, and in maternal serum. And here we have the concentration, the proportion of selenoprotein P in yellow, followed by selenoalbumin, glutathione peroxidase, and selenometabolites. And if you see these graphs, you can observe that it seems that the missing proportion of selenoprotein P that is present in maternal serum could be transferred by human milk. So human milk is an important vehicle of selenium transfer, especially considering that about 90% or 70% of mothers are below the required levels of selenium. And in fact, the concentration of selenium in the maternal serum is higher than in human serum and than in cord serum. And in this other work, we determine metals in human milk of mothers with iodine deficiency. And iodine deficiency was determined spectrophotometrically in urine of the mothers, and then we determine the metals in the human milk. So you can see here in orange the concentration of the elements, and in green the concentration of the elements in the human milk of iodine deficient women. So, for example, regarding selenium, we can see that the concentration is significantly low, and it was expected because, as I commented before, selenium-containing enzymes are involved in thyroid metabolism. And for example, for copper, copper stimulates the T4 production. It is involved in the thyroid metabolism. And also, there is a class of metabolites, which are the phospholipids, which influence the stimulation of the thyroid hormones, and they require copper for the synthesis. So copper is indirectly involved in the thyroid metabolism also. And regarding zinc, zinc controls the activity of thiotrophine releasing hormone, and also the iodination of enzymes and thyroid stimulating hormone. And also zinc transporter have been determined in thyroid gland, but their role still remains unclear. And regarding the metabolomic approach, first we have to select the model organism. After that, we should select the sample. And the first step is the extraction of metabolites. And after that, we can analyze the samples and target, and we can use the mass to charge ions obtained in the spectra to try to classify our sample. And after a discriminant analysis, if the classification is possible, we can look for those ions that are responsible for this classification, for this separation. And we can obtain also the variable importance on the projection, and we can select those masses that are very important to discriminate the groups under study. And after that, we can identify these masses with metabolomic databases or tandem mass spectrometry. And after that, we can perform biological interpretation. In this slide, you can see the results of the metabolomic analysis to the human milk samples that I commented before for metas. And here we determine the metabolites. So here you can see the typical chromatogram for control samples and the typical chromatograms for the human milk of iodine deficient women. So we can see important differences here in the region of diacylglycerols and glycerophospholipids, which are different, and also for triacylglycerols here, or also monoacylglycerols and fatty acids. And here we can see the chromatograms for negative ionization mode, and in the upper part for the positive ionization mode. So the chromatograms are different. We have a different composition. And in the upper part, you can see the discriminant analysis using gas chromatography with mass spectrometry and HPLC-QTOF in positive and in negative ionization mode. And the important thing is that we can separate both groups using metabolites as variables. And in this slide, you can see a heat map of abundance of the different metabolites that we determine to be significantly different between controls and human milk of iodine deficient women. So here in green, we have the controls, and we can see that, for example, this band of metabolites decreases because it is marked in blue in iodine deficient women, and this band increased because it is marked in red. So finally, we identify 31 metabolites with important biological roles, for example, glycerophospholipids for neurodevelopment, that were altered in the human milk of iodine deficient women. And the main metabolic pathways were the lipid metabolism, the amino acid cycle, the T-carboxylic acid cycle, and the glycolysis. In this graph, we can see the metabolic pathways that are altered in the human milk. And for example, the size of the circle means that there is an important impact regarding, for example, the number of metabolites that are altered in these pathways. And the color in this axis means that the impact is very significant. For example, the red color in the upper part means that the p-value is very high. So the lipid metabolism, especially the glycerophospholipid metabolism, the citrate cycle, and the pentose phosphate pathway are present the highest number of altered metabolites in the human milk of iodine deficient women, and otherwise few metabolites belong to the arginine biosynthesis, but the significance is the highest of this pathway. And in this other study, we determined metals in the human milk of mothers with COVID. And in this case, you can see here the discriminant analysis. In green, we can see the control pre-pandemic samples, and in red, we can see the human milk of mothers with COVID. And here you can see the concentration of the different elements. In general, most of the elements, selenium, nickel, iron, copper, vanadium, or aluminum, were found in significant lower concentrations in models with COVID compared with the control, except zinc and also some minor elements like thallium or arsenic. And for example, selenium, you can see here that selenium concentration decreases in the human milk of models with COVID. And there is an important study of Professor Margaret Ryman from the University of Surrey in London, and she demonstrated that in those parts of the world in which selenium concentration was lower in the environment, the severity of COVID was higher, and also that these people have a lower concentration of selenium in the human serum. And also we obtain similar results. The selenium concentration in the human milk of models with COVID is also lower. And regarding copper and zinc, this is not the same. There is another study of Professor Richard Lovinsky of Po in France and co-workers, and they demonstrated that copper levels in serum increase because they are related with the inflammation, and zinc decreases because it has antiviral activity, and in fact zinc has been pointed out to be efficient against COVID management. However, we obtain the opposite behavior in the human milk. Copper concentration decreases and zinc concentration increases, just the opposite than in human serum. And also we calculate the ratios between elements, and only the copper to zinc ratio and selenium to zinc ratio were significantly reduced in the human milk of models with COVID. And in fact, the copper to zinc ratio has been pointed out as an efficient biomarker in lung cancer. And this observation is different in the human serum of models with COVID. So in the human milk, this ratio decreases, and in the human serum, this ratio increases. And also the selenium to zinc ratio was an efficient biomarker combined with selenoprotein P, zinc, and the age. And also this is very important because support the hypothesis of the role of selenoprotein P in redox homeostasis in the endoplasmatic reticulum stress, and also in the inflammatory and immune signaling, and of course in the virus replication. And here we can see the results from our metabolomic analysis. You can see here the discriminant analysis using metabolites as variables. And in this case, we can separate in green the control pre-pandemic human milk samples from models with COVID in red, symptomatic, and models with COVID asymptomatic. It was not possible with metals, using metals as variables, but using metabolites, we can discriminate these three groups. So this is very important. And after the metabolomic analysis, we identified 9 metabolites that were downregulated in models with COVID compared with controls, and 11 metabolites were upregulated in the human milk of models with COVID. And here we can see the graph for the pathways, and the most altered metabolic pathways were the amino acid tRNA biosynthesis here, the phenylalanine tyrosine and tryptophan biosynthesis, and the linoleic acid metabolism. And in this other graph, we can see a heat map showing the associations of metabolites in the human milk with metals in the same samples. This is a heat map for correlation. This is not for abundance as the previous one I showed you. So you can see here two groups of metals. The first group include selenium, copper, iron, vanadium, and these elements are positively associated with this band of metabolites because they are marked in blue. And the same metabolites are negatively associated with the same band of metabolites because they are marked in red. And in the upper part, you can see that this group of metabolites are negatively correlated with this band of metabolites, and these metals, group 2, are positively associated with the same band. And also, the first group of metals present a highest number of significant associations. The stars means that the p-value is below this level, so it is significant. So this group of metabolites are significantly related to a wide number of metabolites in the human milk. And also here you can see also correlation heat maps that show the associations between metals and antibodies of COVID in the human milk, because these samples were used for micro workers in a previous study to determine the COVID antibodies in the human milk. And we determine metals in the same sample, so we can look for associations between metals and antibodies. And here you can see that higher levels of immunoglobulin A here are associated with lower levels of selenium and cobalt, while higher levels of immunoglobulin G here are associated with lead and nickel. So also interestingly, there were no correlations between metals and antibodies in asymptomatic models. Only in symptomatic we found associations. And also, there were not significant associations between antibodies and metabolites in the analyzed samples, only between metals, not with metabolites. And in this slide, you can see associations between, in healthy models, between selenoproteins and microbiota of the human milk. So, for example, you can see that all chemical forms of selenium are significantly associated with this bacteria, lactic acid which is a producer of lactic acid. It is used for functional food and improves digestion and tolerance to milk. And also, several forms of selenium are associated with bifidobacterium, that is also very important. And regarding the gut microbiota of the newborn, we can look also for associations between selenium species and microbiota of the gut content of the newborn. And also we found associations, for example, with bifidobacterium here. And in this slide, I want to show you the results obtained for mice studies that we also developed. And in this study, we used 40 mice divided in 4 groups. Here we have the control group and also mice after supplementation with selenium. And we combine these model mice with another model mice in which we partially eliminated the microbiota with a cocktail of antibiotics. And here we can see that this group is fed a regular diet, and this group was supplemented with selenium. And in this slide, you can see the microbially produced metabolites by the gut content, the microbiota, and the different groups under study. This is the group with microbiota partially eliminated with antibiotics, and this is the normal model mice. So we can see that this is an abundance heat map, and we can see here a band of metabolites that increase here in red, that increase after selenium supplementation, and this band decreases after selenium supplementation. And also with the other model mice, we can see here a band that increases and a band that decreases after selenium supplementation. So this was the first study about on-target gut metabolomics after a microbiota depletion and selenium supplementation, and we determined that about 70% of the metabolites uh, change after selenium supplementation in this model mice, and only 30% of these supplemented mice were different to the control group, suggesting an important effect of selenium supplementation in the microbiota of the mice. And in this other study, you can see a model map showing the concentration of the selenoproteins in the different mice group. Here is the control group. And as expected, after selenium supplementation, we have an increase in the selenium availability and an increase of these proteins. It was expected because we are supplementing the mice, so we have higher levels of selenoproteins in the serum. And here, if we feed mice with rodent diet, not supplemented, but we partially eliminate the microbiota, we have also an increase in the selenium availability. And this is very important because there is a competition between the host and between the microbiota for the selenium. In fact, about 1/4 of all bacteria express selenoproteins and therefore need selenium for the optimal growth. And finally, we conclude that selenium supplementation modulated plasma selenoproteins and all is influenced by microbiota disruption because we have an interwin mechanism. And selenium impact in the microbiota diversity, richness, and also increase the relative abundance of some health-relevant taxa. For example, the well-known lactobacillus, which has been many times related with aprobiotic activity. And in this other work, we try to isolate selenoproteins from brain tissue after the supplementation with selenium in these mice. And we use a typical extraction region for a proteomic protocol. But here you can see the chromatogram obtained. But unfortunately, with this protocol, we can only extract 30% of total selenium present in the sample. So we decide to apply transcriptomics. And here you can see the complete transcriptional profile of the mice brain in the different groups in different colors. For example, this is the control, and this blue is mice after microbiota elimination and selenium supplementation. And the letters here means the significance in the comparison between the different groups. So we detected significant differences in glutathione peroxidase, especially in GPX1 and 4, and also in thyroredoxine reductase, also seleno-R, and for example, also in seleno A here, which is very important for phospholipid homeostasis, and also in seleno T here for maintaining the functionality of dopaminergic neurons, and also in the thyroid hormone metabolism in the diodes, which are here, and we detected also significant differences. And in this other slide, you can see the results of the metabolomic analysis in brain tissue of these mice. We can see here a band of metabolites that increases after selenium supplementation, and here band in blue that decreases after selenium supplementation. And the main important thing is that lipids are the major constituents of brain, and they are completely altered in our mice. So selenium impact in the lipid metabolism of the brain, and also microbiota has an important impact in these metabolites. So we identified 31 metabolites that were significantly altered in the brain after selenium supplementation, and many metabolites were associated with the gut-brain axis, demonstrating a connection between the gut microbiota and the brain of the mice. And also, we determined several important pathways that were altered after selenium supplementation. Here we can see the main ones. For example, the phosphatidylinositol signaling system and other pathways that are significantly altered in red or with a high number of metabolites altered. And in this slide, we can see the associations between metabolites in the mice brain and gut microbiota here. And in this other one, the selenotranscripts in the brain with the microbiota, and here metabolites in the brain with selenotranscripts. So the important thing is that we have many significant associations between them and intertwined mechanisms. For example, cholesterol is associated with these two selenoproteins, seleno-N and thyroredoxin reductase, and these two selenoproteins are associated with these three bacteria in the gut content, and these three bacteria are associated with cholesterol. So we have many connections between them, and several bacteria has been previously reported to be associated with, for example, roseburia, major depressive disorder, or anorexia nerviosa, etc. So they have important neurological functions. And here you can see the results from testicular tissue. We also extract selenoproteins from testicular tissue. Here you can see the typical chromatogram. And for example, this is an advantage of ICP-MS in relation to transcriptomics. We can identify selenoalbumin, which is not a real selenoprotein. In fact, selenium is incorporated into albumin after the synthesis of the protein, so it cannot be identified by transcriptomics. And here you can see a peak showing the variation of selenoalbumin in testicular tissue. And also here you can see the significant changes in the concentration of total selenium in the different mice group after selenium supplementation and microbiota depletion with antibiotics. And here significant differences between the different groups regarding the selenoprotein, selenoprotein P, selenoalbumin, and glutathione peroxidase here. And also, we found important associations between selenoproteins in testicular tissues and gut microbiota. And for example, several selenoproteins in testicular tissue are associated with mucispirirum that has been previously associated to testosterone and sperm activity. And also, for example, with this bacteria that has been previously related with sex hormones in reproductive endocrine system. And in this slide, we can see important results obtained by a photomicrograph showing the cross-section of the testicular tissue. This is the control group. This is the mice after microbiota depletion by antibiotics. And this is the same after selenium supplementation. And we can see here the basement membrane. The basement membrane is essential for animal development. And here in the control, we can observe the membrane. And after microbiota depletion, we can see that the basement membrane has abnormalities. And these abnormalities are prevented by selenium supplementation, almost partially. And also here in this graph, we can see that the microbiota depletion by antibiotics led to a possible arrest of cell division process that convert spermatogonia into spermatozoa here, and this part decreased. And the alteration of the normal structure of these tubes can be in part eliminated by selenium or prevented by selenium. So we can conclude that Omic methodologies for the determination of metals and metabolites, in combination with meta-omic for the determination of the microbiota profile, is a very powerful combination in biomedicine and also environmental issues. And our results open further research related with metals, metabolites, and microbiota in lactating women due to the potential implications for the maternal infant health. Then we suggest to combine metallomics, meta-taxonomics, and metabolomics. And I want to acknowledge my research group of the University of Huelva, especially the young people working at the laboratory, and also my top collaborations, Professor Nieves Abril, that is expert on animal experimentation, and also Carmen Collado from Valencia, which is expert on microbiota. And also the funding, and I want to remember you that Huelva is just here, and this is an important place with a history around Christopher Columbus, and also we have this very nice species, the Lynx pardinus. And I thank you all of you for your attention, and if you have any question, I can ask you. Thank you.